The next talk is entitled Label-Free Analysis and Printing of Cells from Droplets as Individual Colonies from Water and Oil Emulsions, be presented by Emmanuel Mavrakis.
Okay. I'll be talking about the work that I've been doing in the lab lately, and this work concerns the development of a technique that can enable label-free analysis and printing of cells from droplets as individual colonies from water and oil emulsions. So it's essentially a technique that can both permit detection, screening of cells for the phenotype, and isolating these cells, capturing these cells. And phenotypic-based screening of cells gets used in various applications from enzyme engineering, synthetic biology, monoclonal antibody production, or antibiotic resistance, to name a few. But our focus has been towards contributing to the advancement of enzyme engineering for biocatalysis. And that's because biocatalysts demonstrate apparent advantages over their synthetic counterparts in that it has allowed us access to the production of molecules in an efficient, selective, and sustainable way. And if we consider the range of possibilities that enzyme engineering has opened to us, the range of possibilities that it has brought to the table, now we can engineer enzymes. We can tailor them to substrates of interest so that they can catalyze reactions to synthesize products that are of interest in food, chemical, and pharmaceutical industry. And one such example is the manufacturing of this anti-diabetic drug, sitagliptin, that was reported by Merck in 2010. So they started with an enzyme with only a marginal performance for the production of chiral amines, and they evolved it. And in doing so, they performed this biocatalytic synthesis of the product, and in doing so, they circumvented the use of this synthetic rhodium-based catalyst and bypassed another synthetic step in order to efficiently manufacture the product. Now, the way they evolved this enzyme was through directed evolution, and one such workflow of the directed evolution is illustrated in this right-hand corner of this slide. So basically, it starts with a single gene, then it codes an enzyme, and then through random mutagenesis and error-prone PCR, one can end up with a myriad of different genes that express variant enzymes. So after these are expressed, there are mutated enzymes isolated in the wells. You can add substrates and have reactions carried out in each of these wells. And then at this point, you need a high throughput screening technique in order to go through all the samples that amount to 100,000 or millions of samples. And that's the bottleneck of these campaigns. That's because the analytical techniques that go through these well plates and analyze these well plates cannot keep up with the throughput requirements. One way to do it and meet these speed requirements is through the use of droplet microfluidics, and there have been reports of the use of droplet microfluidics for directed evolution. And in this part, what you do is you encapsulate cells into aqueous droplets within a flow of oil. And then if, for these reports, the substrates were fluorescent, the products were fluorophores as well. So you can use fluorescence readout, and you can process many of these droplets in a rapid fashion. However, fluorescence is limited in terms of the scope of analytes that you can target. So a way to do it is through label-free screening, and that's through the use of mass spectrometry. And if you combine droplets with mass spectrometry, you get tremendous advantages with respect to analytical costs and speed. So that's the way to do it. And that's what we set out to do. And another feature that you would want for a droplet technique is the ability to reclaim all these droplets that you have interrogated the phenotype of the cells that were housed in these droplets, right? So a way to do it would be to reclaim these droplets and retrieve the cells out of these droplets, and this would streamline the directed evolution campaigns in terms of gene sequencing and all these time-consuming steps that would be needed if you had a technique with droplets that would pull all these droplets in a vial. So that would be increasingly difficult to take the cells out of that and get to the level of single colonies. So that's what we set out to do, integrating droplet MS with a technology that can capture these interrogated droplets. And this is a graphic representation of how we implemented this idea. We are starting from encapsulating our cell populations into these droplets in a very fast manner, and we can pump all these droplets into a storage vials where they can stay for a appropriate amount of time to allow for the incubation, for conducting these reactions. And then from these reservoirs, you can pump them into a microfluidic chip that is a re-injector and splitter device. And here you can see we've pumped these droplets in this zoomed inset, so the droplets are waiting here in this channel to get re-injected into this main channel by a complementary flow of oil, of a microfluidic oil that's typically used in making these emulsions. So the droplets, they will get into this channel. And what you can see here is at the end of this channel is split into two branches. So a droplet that travels through this channel, it will split into two fractions. One fraction now, you can route it through a natural electrospray source. That's a sheath spray source that works fantastic with respect to droplet detection. And the other fraction, the sibling fraction of this droplet, you can route it through a custom voltage-mediated deposition probe that we have made in the lab in order to collect these droplets on agar plates, a growth medium for the cells that are contained within droplets to grow. And here you can see a robust operation of our system. We can process these droplets rapidly, and it will produce a symmetric splitting of these droplets across a range of reinjection rates from 0.4 to 1.2 droplets per second. We've worked with 15 to 20 nanoliter droplets, and the system is pretty robust for a statistically significant number of droplets, and can run unproblematically for large sessions. Here I'd like to show you how the voltage-mediated droplet pull-down works, our technique. So here this is a cycle for one single droplet that's getting pulled down onto the deposition substrate. So what we have is a HPFA capillary that's dedicated to transferring the droplets, the sibling droplets from the microfluidic chip. And what we have done here is we have threaded it into a metal capillary. And here you can see another metal plate which is hollow. It has a hole below the level of this capillary. What we're doing is we're applying an electric field between these metal objects. And what you're going to see in this video is what this electric field causes. It causes the oil to be issued as a thin jet and be sprayed down out of the emulsion, while the aqueous droplet is gently, smoothly pulled down without compromising its integrity. And this is happening because of the considerably larger surface tension of water. What's keeping it compromised? That the oil can spray because of its low surface tension. However, the water phase would need twice as much voltage in order to fragment it and convert it into an aerosol. But at the voltages that we operate our system at, we do not see that. And now that we have this voltage-mediated probe, you can very nicely mount an agar dish, as you can see here in this picture. You can mount an agar dish below this printing probe in order to collect these droplets. And this dish is mounting out on a XY, a two-dimensional moving stage. So droplets are continuously pulled down from the probe, and they will land softly and stick on the agar. The agar is continuously moving because it stays onto the plate, so we're printing at a pattern that is predetermined by our programmed translational stage. So you can see the droplets rapidly sticking. And we wanted to test the idea of simultaneous, synchronous detection of our droplets and what happens when we print them onto the plate. So we devised an experiment where we had mixed populations of cell-containing droplets. One population contained E. coli that were expressing the green fluorescent protein, and another population contained the red E. coli that were expressing the red fluorescent protein. And we added dopants, electrospray molecules that could be easily detected by the electrospray. So we knew that whenever we detected the dopant, it would correspond to the RFB or the GFB cell. And here you can see the droplets getting detected as distinct bursts in the recorded signal. So you can see the trace here. However, what happens when the droplets get printed onto the agar dish? They grow into colonies. So the cells, they grow colonies, and because they are fluorescent cells, you can image the plate afterwards, so you can get distinct characteristic traces, green traces for the GFP E. coli and red traces. And if you align the pattern of these fluorescent signals with a pattern of signals that you detect with the MS for the dopants, you can get very excellent high degree of alignment. The lowest has been 94%. So this shows that the location of the spot here can be mapped back to the mass spectrometer signal, and that's the phenotype that you have interrogated for this cell. So it shows a way forward for this technique to contribute to directed evolution. And here is the recognition of the signals. Now, I wanted to test this technique in a real scenario, in a real screen scenario, and one good candidate was lysine. And lysine is an essential amino acid that's used in feed additives and dietary supplements, so there's $1 billion demand there. And because of that, they have been set up campaigns in order to evolve enzymes, microbes, in order to produce lysine. And to have this campaign, in order to conduct this campaign, 16,000 variants could be screened with the LCMS, and that would take 34 days to complete this evolution cycle, whereas with droplets it would take 9 hours. So this was a good case to test our technique, evaluate its strengths. And we did this experiment. We mixed populations of E. coli producing, lysine producing E. coli, this strain, with control cells that were used both for controlling the levels of lysine and also as markers for the deposition. So the control cells with RFB E. coli, they were not supposed to produce any lysine. What we could detect were only basal levels of lysine. So we introduced these droplets with our online system, and we can see that we can conveniently discriminate between the two populations of cells, controls and lysine producing cells. And we collected all these about 800 droplets. And so when you inspect your agar dish, lysine producing E. coli would have no fluorescent signal. They would only be visible in this bright field micrograph, whereas the red fluorescent traces, that would be visible, and then you can see if your pattern matches with the mass spec, and it matched nicely. And a little bit about the droplet printing capabilities and the throughput capabilities that our technique can offer. Here you can see an agar dish that is beautifully printed with colonies, and we can achieve millimeter to sub-millimeter precision when printing with our system. This is a plate accommodating 400 droplets, printed at 1 droplet per second. So we're very happy for that. And a little bit about the improvement that we can get over well plate counterparts and a rapid screening technique based on well plates. Typically for an evolution cycle of 25,000 variants, with a well plate LCMS screen, you would take, this is expressed in 10-hour workdays. It would take almost 120 days to complete this evolution cycle. Whereas one faster report of this system with solid phase extraction reports a faster throughput, but still this faster throughput does not compare, it's not near the workloads that we can have with our droplet MS techniques. You see here at the faster rates, we can complete this evolution cycle within a day. I'm wrapping it up. So at this point, I'd like to say that we demonstrated for the first time an online MS-based cell phenotyping and direct capture in agar. To the best of our knowledge, there's not a single technique on that. We demonstrated a novel deposition approach that can print onto any substrate regardless of the material of the substrate. We can achieve convenient alignment of colony position with cell phenotype, and that means that the colonies can be easily picked up to get forwarded for gene sequencing at a later step, a step that is required in directed evolution. It's a versatile system that can be interfaced onto any electrospray MS source, and it can also be interfaced to atmospheric pressure chemical ionization. So in this way, you extend your a directed evolution range to non-polar molecules, like terpenes, for example. And we also envision additional applications for this system in clone selection for monoclonal antibody production, and we also see applications for rare cell capture in mass cytometry by use of ICP-MS. At this point, I'd like to express my thanks to my advisor, Robert Kennedy, and all the Kennedy Lab that has hosted me in this postdoc. Particularly, I'd like to thank my colleagues, Thomas Knecht and Maxwell Unger, who have actively contributed to the advancement of this technique. Of course, the Narayan Lab for their support in this project, NIH for funding, and last, we would like to thank Agilent for sponsoring our single quad mass spectrometer that has been the workhorse for this project. It's extremely robust. Sometimes I think you can throw it out of the balcony and it will still work. And thank you all for your attention.
Let's not test that last slide. Okay, we have time for questions.
Two quick questions.
Yeah.
First, really interesting, nice talk. Just quickly, in the beginning, how do you, when you're collecting in the reservoir your droplets, how do you keep them from coalescing? Is that a problem? It seems like that should happen.
You use surfactants, so you decrease the surface tension, and in that way you can keep the emulsion stable.
Okay. And then, I don't know if this is related, but in your printing, your pattern of the droplets on your agar plates, it seems like there was some random variation in terms of the spot position, even the droplet size. Is that because of instability of your voltage source that you're applying to do this, or where does that variation come from in terms of?
In terms of the size of the spot?
The size and the position of the spot.
So the position, there is some variation. It's quantitated at the millimeter scale. The faster you go, the variation increases a little bit, but this doesn't prevent us from picking the colonies because the rows that at which we print is 5 millimeters. So the outlier, the largest variation that I've seen, it's about 1.5 millimeters. So I understand that there is some variation and it's not a perfectly straight line. And there might be various sources of error contributing to that. Now about the size of the spots, usually there might be variation in the size if your cell concentration in the droplet is not uniform across all droplets. So you have a lot more starting material, more cells thrown down, and this will will develop into a larger colony compared to a smaller cell concentration.
All right. Thank you very much, Manos.
Cool.